So class, let's talk about glycolysis. It's actually one of the first metabolic pathways that we studied in this course. So I thought I'd go over step by step the 10-step pathway that cells use, both aerobic and anaerobic cells utilize to catabolize glucose. So catabolize meaning that glycolysis, lysis of glucose. So this is a way that the cells break down glucose to get energy, and that energy usually comes in the form of ATP. So this is a 10-step reaction pathway. It is a metabolic pathway, so it is subject to flux. It is subject to shunting in and shunting out. And all metabolic pathways are subjected to regulation. Allosteric regulation, feedback inhibition. So it's just a way to modulate how fast a cell is to undergo glycolysis, or how slow a cell is to undergo glycolysis. A lot of this regulation occurs based on regulation, allostery, but it also occurs at a higher level, something we really didn't mention in class, but hormones can also regulate the flux through glycolysis, specifically the hormones insulin and glucagon as well. So that is at a higher level of organization. Let's first talk about the net reaction of glycolysis. So first, glucose, usually by dietary intake, will react with 2 NAD+, 2 oxidized form of the nicotinamide adenine dinucleotide cofactor, plus 2 molecules of ADP, plus 2 molecules of inorganic phosphate. Okay. So this reaction is one direction, though some of the sub-reactions can go in both directions. So they will yield this NAD+ will become NADH. This ADP and inorganic phosphate will react to form 2 ATP, so that's a good thing. Okay, any metabolic pathway that generates reducing equivalents in the form of NADH is good, because those reducing equivalents can, if it's an aerobic organism, donate those electrons, those reducing equivalents, to the electron transport chain. And obviously ATP is also good to have. And then this glucose actually goes to pyruvate. And there is a 4 protons are given off, and an H2O is given off, 2 H2O. Okay, actually you produce 2 pyruvate for every 1 molecule of glucose that enters into the glycolytic pathway. So here is glycolysis. As stated before, this does not happen in one step. This is a 10-step process. So metabolism splits the net reaction into any number of steps. In this case, it's 10 steps. And we discussed the advantages of doing that. One of the advantages is that you can create high-energy molecules, and then you can create intermediates that could be shunted to other pathways or other networks in the cell. Also regulation. Regulation means that you can slow things down. It's really hard to slow down one reaction, one net reaction, but if you split this net reaction into 10 individual sub-reactions, you can regulate at those sub-reactions. Most regulation occurs at the rate-limiting step of the enzyme, or the rate-limiting enzyme, rate-determining enzyme. And glycolysis, that enzyme, is phosphofructose kinase. The enzyme that really commits the cell towards this particular pathway. So here is glycolysis. Glucose gets converted to 2 molecules of pyruvate. Let me just draw the structure of pyruvate here. Okay, pyruvate is going to look like this. And glucose, we know from our lectures on carbohydrates, is going to look like this. Okay, so it could be the alpha form or beta form. I'm not going to really worry about that. So I'm just going to draw the beta form of glucose here. Or I'm sorry, the alpha form of glucose. But we're not going to worry about the anomers of glucose, whether it's an alpha anomer or beta anomer. Okay, so here you see the anomeric oxygen. Here's the anomeric carbon. The OH can be below or above the plane of the ring. And here is the molecule that's going to be utilized for a tremendous amount of energy from the breakdown of this 6-carbon skeleton into 2 molecules of this 3-carbon. Actually, it's called an alpha keto acid. And that alpha keto acid is called pyruvate. So if you think about it, we are converting an aldose sugar, an aldose hexose sugar, getting ATP from it, and we're converting it to 2 carbons. This is 6 carbons, so 6 carbons. We're converting it to 2 3-carbon molecules of pyruvate, an alpha keto acid. Now, what is the fate of this pyruvate? Well, it can be utilized to make more energy if it's an aerobic organism, but it's also utilized for some bacteria. It's also really utilized to regenerate the oxidized form of NADH into NAD+, so that this whole reaction can go through another round. So usually the recycling of or regeneration of NAD+ occurs in a lot of bacteria, but aerobic organisms will take this pyruvate and convert it to the TCA cycle and have an entry port for electron transport. All right. So this is what cells do. Bacteria cells do this, and muscle cells and liver cells, brain cells. Okay, so almost a universally evolved and consensus pathway across all facets of life. Okay, plant cells, fungus, you name it. Glycolysis mostly is occurring. There are some form of this pathway. Some textbooks also call this the Ebden-Meyerhof pathway. Okay. The Ebden-Meyerhof pathway is also another name for glycolysis. So let's go over all 10 individual subreactions of glycolysis. Adding all 10 of those reactions will give you this net reaction of glycolysis. So the first reaction of glycolysis is catalyzed by the enzyme hexokinase. And in this enzyme, what it does, it takes glucose. So let me draw glucose again here. I'm just going to draw the alpha form of glucose. Okay, not worrying about anomers, though you'll have a mixture of alpha, a mixture of the beta anomer. You'll even have a small form of the straight chain aldehyde. Okay, so what happens in the first step of glycolysis in reaction 1, ATP is actually utilized. So you are expending an ATP molecule. Okay, and this is basically priming that number 6 carbon. So if I were to number these carbons 1, 2, 3, 4, 5, and 6, the number 6 carbon gets phosphorylated, and that donor is an ATP molecule. So enzymes that phosphorylate are called kinases. This particular enzyme is called hexokinase. ATP is a significantly negative molecule, simply due to the fact of those 3 phosphates. All of those phosphates have a negative charge. So a lot of times in the cell, magnesium sort of neutralizes that negative charge. Most textbooks don't put magnesium, but magnesium is essential for hexokinase and the first step of glycolysis to work. So this is reaction 1 of glycolysis. And you can imagine what happens here. We're going to have a nucleophilic attack on the gamma phosphate, and ADP will come off, which is fine. But the important thing is that we phosphorylated glucose at the number 6 carbon, and we get glucose 6 phosphate. What I'll do is I'll just put this phosphate in green or orange. Okay, this phosphate P. Okay, this phosphate P is actually PO3 minus 2. So here we have the first reaction in glycolysis. Okay, as catalyzed by hexokinase, glucose gets converted to G6P, glucose 6-phosphate. Couple of words on hexokinase. Couple of words on hexokinase, the enzyme that catalyzes reaction 1 of glycolysis. We have several forms of hexokinase. A form in the liver is very specific for glucose. So if you do the CHEM, the Michaelis constant, it's going to be very, very low and have a very strong affinity for glucose and glucose only. But other cell types will have a hexokinase that is very promiscuous. Okay. Their hexokinase HK is very indiscriminate or promiscuous. By promiscuous, it can phosphorylate not just glucose, but mannose, fructose, other types of aldose and hexose sugars. Okay, so that's the first reaction of glycolysis. We bring the glucose in. Glucose gets transported. We usually have a lot of transporters in the cell. The most famous transporter is GLUT. GLUT1, GLUT2, GLUT3, GLUT4. The GLUT family of transporters bring glucose in. And hexokinase will work in phosphorylating the number 6 carbon. Hexokinase has a Pac-Man, very classic induced fit conformational change. All right. Let's go to reaction number 2. We take this glucose 6-phosphate. Notice we've expended an ATP molecule here, so we can, if we want to do a bank balance, we have removed an ATP molecule from the cytoplasmic pool. So that's reaction 1. The second reaction of glycolysis just takes that glucose 6-phosphate and isomerizes that into fructose 6-phosphate. So that is an aldose-ketose isomerization. So the textbook has the mechanism of how you go from an aldose to a ketose. I'm not going to belabor that point. I'll just draw the structures of reaction 2 of glycolysis. So here we have glucose 6-phosphate from reaction 1. I will draw that as the substrate now, the continuation for reaction 2. So here's glucose 6-phosphate. I'm just going to draw the inorganic phosphate as P with a circle, okay, from here on out, but just realize that P with a circle is PO3 minus 2. That's what it symbolizes. So here's glucose 6-phosphate from reaction 1. From the hexokinase reaction. And we will undergo an isomerization. Okay? So here, in this particular case, this reaction is actually reversible because going from an aldose to a ketose actually is usually base catalyzed or an acid catalyzed reaction. Once again, if you want, you can refer to the textbook. Also, the PowerPoint notes has the mechanism. But a base catalyzed reaction splits open this ring, and then the ring closes so that you get the ketose form. All right, so let me draw the ketose form here. This is the ketose form of the sugar, a 6-carbon sugar, and that is called fructose 6-phosphate. So everything else has remained the same. Again, that phosphate in the circle is PO3 minus 2. So this is fructose 6-phosphate. Okay? Remember, here's the anomeric carbon in ketoses. Here is the anomeric carbon in aldoses. And the carbon numbering is 1, 2, 3, 4, 5, 6. The carbon numbering here is 1, 2, 3, 4, 5, and 6. All right. So the enzyme that catalyzes this reaction that exists in equilibrium, so it really goes back and forth, so you can adjust the flux accordingly. The enzyme that does this is called phosphoglycoisomerase, or PGI. Okay, this is one of those enzymes that's implicated in hemolytic anemia. A lot of enzymes in glycolysis are associated with some form of anemia or the other. You may remember glucose 6-phosphate here can go into the pentose phosphate pathway, and in that pentose phosphate pathway you produce NADPH, and you produce other molecules such as fructose 6-phosphate, glyceraldehyde 3-phosphate. You also produce ribose 5-phosphate for DNA synthesis. Okay? And you also produce NADPH. So, the shunting of glucose 6-phosphate into this particular pathway, the pentose phosphate pathway, really directs the cell to go more along the lines of reductive biosynthesis. Okay? And, one of the major enzymes that takes glucose 6-phosphate and converts it to the carboxylic acid, the gluconate form, is glucose 6-phosphate dehydrogenase. And that enzyme also is implicated in hemolytic anemia, just as phosphoglycoisomerase. So you're going to see hemolytic anemia a lot as one of the main pathologies associated with defective enzymes, particularly in glycolysis and the pentose phosphate pathway. So this reaction too is catalyzed by phosphoglycoisomerase, and it is an aldose ketose isomerization. Okay? Remember, fructose and glucose, and by extension glucose 6-phosphate and fructose 6-phosphate, are isomers of each other. They're both 6-carbon sugar molecules. So that's the second reaction of glycolysis. Let's move on to reaction 3. In reaction 3 of glycolysis, we have the fructose 6-phosphate that we generated from our second reaction, our fructose 6-phosphate, and we're actually going to phosphorylate now on the number 1 carbon. So here is an expenditure of ATP in our third reaction of glycolysis, where we'll expend a second molecule of ATP and generate fructose 1,6-bisphosphate. That bis just basically means that you have 2 phosphates on 2 separate carbon atoms within one molecule. So let's go over our third reaction. It's actually a very important reaction in terms of regulation. So here's our fructose 6-phosphate. Once again, that is fructose 6-phosphate. We will utilize one ATP molecule here, a second ATP molecule. Notice the direction of the arrows. Okay, so the arrow is going in one direction. The enzyme that catalyzes this reaction, or reaction 3 of glycolysis, is phosphofructose kinase. Okay, also known as PFK1. And PFK1 is going to take that ATP molecule. Okay, this does a nucleophilic attack on the gamma phosphate. ADP comes off as one of the products. Okay, don't forget, magnesium is very important in chelating those negative charges of the phosphate. And then as a result of this, you will get ADP plus fructose 1,6-bisphosphate. So CH2O, put in another phosphate here. I'm going to put it with an orange circle. It's the same thing here. And let me just complete the structure of fructose here. The rest of the 6-carbon ring, or 5-carbon ring, excuse me. So here's reaction 6 of glycolysis. Okay, fructose 6-phosphate from reaction number 2, the ketose form with ATP. So if we're going to do our bank balance, we have now 2 ATPs, one from hexokinase and the other one from phosphofructokinase, that donates its phosphate group. This is a nucleophilic attack on the relatively weak hydroxyl group on the gamma phosphate. ATP is a high energy molecule, so ADP serves as the leaving group, and you have fructose 1,6-bisphosphate. Okay, fructose 1,6-bisphosphate. All right. This is a molecule that is ready to go to reaction 4 of glycolysis, but it's worth mentioning or taking a few moments to recognize the importance of phosphofructokinase. It is the rate-committed enzyme in glycolysis, and it's heavily regulated. Okay, so there's a lot of allosteric factors that will change the flux and activity of this enzyme, and by virtue of that, change the quantity of fructose 1,6-bisphosphate that can go into glycolysis. So rate-determining enzyme, rate-committed enzyme, bottlenecked enzyme, that is what phosphofructokinase, or PFK1, is. By the way, the reverse enzyme that catalyzes the reaction in the opposite direction, so since this reaction goes in one direction, there's a separate enzyme that catalyzes the reverse reaction, and that is fructose 1,6-bisphosphatase. Fructose 1,6-bisphosphatase. Okay, so that's a separate enzyme that's going to cleave off the phosphate to give you fructose 6-phosphate. So if a fructokinase phosphorylates this substrate to give you bisphosphate, then a bisphosphatase will cleave one of those phosphates to give you back the original molecule. So separate enzyme that goes this direction, separate enzyme that goes in the opposite direction, 2 different enzymes, 2 different genes that code for those enzymes. And these enzymes are very sensitive to allostery, and these 2 enzymes are very sensitive to hormones, glucose, very sensitive to hormones glucagon and insulin. So in later courses, you'll actually study how hormones, insulin and glucagon, and maybe epinephrine, a couple of other these hormones, regulate at a higher level the activity of these enzymes, and henceforth the flux through the pathways. But besides regulation at the hormone level, you also have allostery. ATP, citrate, ADP, ratios of NAD plus to NADH, the ratios of the reducing equivalents really influence how well this enzyme functions and particularly flux through the pathway. All right, a very, very important enzyme. It's definitely worth your time and effort to study this some more, especially if you want to go into higher other fields in science. But for now, I'm going to move on to reaction 4 of glycolysis, and that is taking that fructose 1,6-bisphosphate, and we're just going to cut that molecule. We're going to cut that molecule in half, and the enzyme that does this is called an aldolase. We do an aldose cleavage. So for reaction 4 of glycolysis, we're going to cut the molecule right across this plane, okay, and get 2 molecules. Each molecule is 3 carbons. All right, so that's reaction 4 of glycolysis. So let's do this again. Fructose 1,6-bisphosphate is going to be the substrate for reaction 4 of glycolysis. So bear with me. Appreciate your patience as I draw the structures, but I think drawing this out should be good to see where stuff goes and how stuff enters and leaves this pathway, glycolysis. All right, fructose 1,6-bisphosphate. And the next step of glycolysis, as I stated before, we're going to just cut this molecule down this plane, okay? The enzyme that does this is called an aldolase, and the aldolase actually runs in both directions, so you can have an equilibrium. If you split this molecule along this plane, what happens is that one of the molecules, as you split it, becomes a alcohol, and this side, and one of the molecules at this end, the right-hand end, becomes an aldehyde. Okay, so we actually get two molecules. We get an aldehyde, and then we get a phosphate. Okay, I'm sorry, an aldehyde, and we get an alcohol. Okay, so cutting this molecule in half, okay, one molecule will still have the phosphate, the other molecule will still have the phosphate, and what we get is glyceraldehyde 3-phosphate going this way, and dihydroxyacetone phosphate going that way. So let's draw the structures of these. All right. So let me first draw the structure of dihydroxyacetone phosphate, DHAP. Okay, so here is the CH2, here's my phosphate. Dihydroxyacetone phosphate. Acetone phosphate, dihydroxyacetone phosphate. So that is going to be the structure of DHAP. Okay, dihydroxyacetone phosphate. And then the other one will be the aldehyde, glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. So dihydroxyacetone phosphate, glyceraldehyde 3-phosphate. Okay. So I'm not going to go over the mechanism of this aldolase reaction, but suffice to say that we have just cleaved the molecule in half. Okay, and one half picks up, so this CH2O phosphate picks up and becomes dihydroxyacetone phosphate, and this CH2O phosphate on the left gets cleaved, and that becomes glyceraldehyde 3-phosphate. All right, so that is the reaction catalyzed by reaction 4 of glycolysis. Okay, the aldolase reaction. We cut this ketose into two molecules, each of them 3 carbons. This one is dihydroxyacetone phosphate, that's 3 carbons. Glyceraldehyde 3-phosphate, 1, 2, 3, that's 3 carbons. This thing is 6 carbons, right? 1, 2, 3, 4, 5, 6. If you want to continue the tracking of carbons, this is going to be carbon 1, 2, 3. That was 1, 2, 3, and then 4, 5, 6 becomes 4, 5, 6. If you're just tracking along the carbons. Okay, so 6 carbons get split into one 3-carbon molecule and another 3-carbon molecule. So we still have 6 carbons. So this is reaction 4, and the reaction is catalyzed by the enzyme aldolase. Okay, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Enzyme is aldolase. So now let's go to reaction 5 of glycolysis. Okay, so in reaction 5 of glycolysis, what we actually do is we take all of the dihydroxyacetone phosphate and convert it to glyceraldehyde 3-phosphate. So aldolase converts one molecule of fructose 1,6-bisphosphate to one molecule of this and one molecule of G3P. The next enzyme converts that one molecule of DHAP to G3P. So as a result, at the end of reaction 5, we have 2 molecules of G3P. So reaction 5 is catalyzed by the catalytically perfect enzyme Triose phosphate isomerase. So reaction 5 actually takes the substrates of these two reactions and interconverts them. So dihydroxyacetone phosphate, whose structure looks like this, dihydroxyacetone phosphate will get converted to glyceraldehyde 3-phosphate. Now this enzyme, Triose phosphate isomerase, operates in both directions. It can easily go from DHAP to glyceraldehyde 3-phosphate, whose structure I'll draw here. Glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. It can easily go back and forth, but in glycolytic flux, in order for the next step, for reaction 6 to occur, reaction 5 must all go towards the production of G3P. So the net effect is you have 2 molecules of G3P that are generated. Okay? So triose phosphate isomerase can go both ways dependent on the flux, but for glycolysis, all of the DHAP goes to G3P. So from here on out, we have 2 molecules of G3P that enter into the glycolytic pathway. Triose phosphate isomerase is also implicated in methyl glyoxal toxicity. It's protective against that based on that random coil secondary structure. Your book has a very nice structure and that deals with it, and we also mentioned that briefly in class. We also mentioned this enzyme is perfect. Has a catalytic efficiency on the order of 10 to the 8th, 10 to the 9th. One of the most efficient enzymes there is, very fast. Once one of these substrates, this or this, gets into the active site, almost immediately the product is formed. All right, so let's go now to reaction 6. We are halfway through glycolysis. We've made 2 molecules of G3P. So from here on out, I'll just do 1 molecule of G3P from here on out. They'll realize from that reaction 6, 7, 8, 9, 10, 2 molecules are actually entering into the pathway. I'm only drawing 1 molecule for reaction 6 through 10. So in reaction 6, we actually take that glyceraldehyde 3-phosphate and we convert it to a high energy molecule. That high energy molecule is called 1,3-bisphosphoglycerate, and that's going to be catalyzed by the very important enzyme glyceraldehyde 3-phosphate dehydrogenase. It's an enzyme that we studied very much in class, and it's an enzyme that we talked about in terms of uncompetitive inhibition. It's an enzyme we've talked about in terms of producing the first reducing equivalent in glycolysis, and that is NADH. So let me draw G3P here. Now we're on reaction 6. So glyceraldehyde 3-phosphate dehydrogenase, as in all dehydrogenases, they are redox reactions. So this one will take NAD+, okay, and it's going to take inorganic phosphate, and this dehydrogenase, glyceraldehyde 3-phosphate dehydrogenase, is multidirectional, or it's bidirectional, so it is in an equilibrium. And you're going to get a good molecule here, NADH. So it's actually 2 NADHs, right? Because I'm only drawing it for 1 molecule of G3P, but thanks to triose phosphate isomerase, we have 2 molecules of G3P that enter in. So we get NADH and we get 1,3-bis phosphoglycerate. So what that means is that we have another phosphate that's going to be attached about right here, okay? About right here in this carbon number 1, or if you want to call it that. And that is a high energy molecule. That is a molecule that is really not stable. That C double bond O phosphate bond is not really stable. So we would expect some stuff to happen in reaction 6, 7, 8, 9, and 10. So NADH plus 1,3-bis phosphoglycerate. Okay. So here's that. So this is where that phosphate goes. I'm going to draw this line as squiggly, okay, because that phosphate is primed to get out of there. It's primed to leave. All right, and this is called 1,3-bis phosphoglycerate. 1,3-bis phosphoglycerate, also known as, well, let me write out the form here. 1,3-bis phosphoglycerate. The enzyme that catalyzes this reaction is glyceraldehyde 3-phosphate dehydrogenase and its reaction 6. GAPDH. Glyceraldehyde 3-phosphate dehydrogenase takes glyceraldehyde 3-phosphate and converts it to 1,3-bis phosphoglycerate. Okay. This is a bond that is labile, meaning it's not very, very stable. It's volatile and could break off any moment. In fact, that's what it does, okay? It breaks off in reaction 7 of glycolysis. In reaction 7 of glycolysis, that 1,3-bis phosphoglycerate donates that phosphate, and you'll get 3-phosphoglycerate. So we count the carbons. This will be 1, 2, and 3. Don't forget that there's actually 2 molecules going through here. Thanks to the triose phosphate isomerase enzyme, there's 2 molecules, so I'm only drawing the reaction for 1 molecule. All right, reaction 7. So after reaction 6 of glycolysis, as catalyzed by glyceraldehyde 3-phosphate dehydrogenase, we go through reaction 7. And in reaction 7, we take the 1,3-bis phosphoglycerate that we generated here, and we, as stated before, it's going to donate this phosphate group to ADP to give you ATP. So this is a high energy molecule. It's one of the 2 high energy molecules in glycolysis. And so that 1,3-bis phosphoglycerate is going to donate this phosphate and phosphorylate ADP to give you ATP, and you will get, as the product after donation of phosphate, we get the product of 3-phosphoglycerate. So let's do that reaction here. Okay, so here we have C double bond O. Here is the phosphate that came from 1,3-bis phosphoglycerate. So here's 1,3-bis phosphoglycerate. It's going to use ADP, okay? And this reaction actually is reversible. And you will generate ATP, so phosphate is going to phosphorylate this. This is a high energy molecule. You'll have ATP. And then this becomes 3-phosphoglycerate. The structure of 3-phosphoglycerate is shown here. The enzyme that catalyzes this is reaction 7 in glycolysis, and that is phosphoglycerate kinase. Kinases phosphorylate things, are enzymes that phosphorylate. So this kinase is going to phosphorylate ADP, the donor being 1,3-bisphosphoglycerate, to give you ATP. Okay? So this type of generation of ATP, this high energy molecule within itself, is called substrate level phosphorylation. Okay, we contrast that with oxidative phosphorylation, which mitochondrial aerobic organisms utilize. So this is a very exergonic reaction. The energy that's released is roughly about minus 65 kilojoules per mole. So a lot of energy that's released. It gets trapped in the form of ATP. And again, this phosphate is a very good leaving group. This bond is actually labile. So this bond is very unsteady. So it would want to leave off, come off, and go and phosphorylate ADP to ATP. Okay? So it may be worth some time to take a little bit of a tangent. By the way, this is reaction 7 of glycolysis. And realize that many cells, particularly red blood cells where glycolysis is almost ubiquitous, many red blood cells have a alternative pathway where they shunt this off and they make the molecule 2,3-bisphosphoglycerate. Okay, the difference between 1,3-bisphosphoglycerate and 2,3-bisphosphoglycerate is just that this phosphate moves from the number 1 carbon to the number 2 carbon, so that's a mutase. So this 2,3-bisphosphoglycerate that's generated in this shunting reaction by a mutase, a separate mutase, can actually shift the equilibrium of hemoglobin from R to T. Okay, so it actually shifts hemoglobin more towards the deoxy state as opposed to the oxy state. Right, so this is an example of shunting. This is why we don't perform the glycolytic reaction in one step, okay? Because we can actually make intermediates that allow for shunting to other important aspects. And in this case, that important aspect is regulating oxygen binding to hemoglobin. So during glycolysis, some of this 1,3-bisphosphoglycerate, if the glycolytic flux is going strong, can be diverted through a mutase, 2,3-bisphosphoglycerate mutase, that generates 2,3-bisphosphoglycerate. A mutase just takes that phosphate and shifts it to another part, to a different region of the same molecule. So it's going to shift it from the number 1 carbon to the number 2 carbon. And that 2,3-bisphosphoglycerate is a allosteric effector of oxygen binding to hemoglobin. So shunting, a common theme in metabolism. We see that in glycolysis. It also guides our attention to the fact that oxygen binding to hemoglobin is somehow intimately related to glycolytic flux. So if flux through glycolysis is high, we can produce theoretically more shunting away of 1,3-bisphosphoglycerate to the production of 2,3-bisphosphoglycerate, which binds to hemoglobin, the 4 subunits, the tetramer of hemoglobin, and causes the conformation to go from the relaxed state to the tight state, in essence facilitating an oxygen dump of the hemoglobin molecule. Okay, you see this common in a lot of areas in terms of acclimation to high altitudes and high mountain areas, or just simply anoxic conditions, low oxygen conditions. Flux some of this towards 2,3-bisphosphoglycerate and change oxygen binding to hemoglobin. So in the red blood cells specifically, glycolysis and binding of oxygen to hemoglobin are intimately related. So let's go now to reaction 8 of glycolysis. Reaction 8 takes that 3-phosphoglycerate and isomerizes it, or actually it's a mutase, converts it to 2-phosphoglycerate. So here's 3-phosphoglycerate from before. Let's draw that again. And in 3-phosphoglycerate gets converted to 2-phosphoglycerate, and the enzyme that does that is a mutase. So again, like before, we have a back and forth. So the enzyme that does this is phosphoglycerate mutase. And a mutase, just like the mutase we saw here, it just takes that phosphate and puts it in a different part of the molecule. Here, this phosphate is going to go from the number 3 carbon to the number 2 carbon. So you get 2-phosphoglycerate. Okay, 2-phosphoglycerate. So by the way, this enzyme, phosphoglycerate mutase, which is reaction 8 of glycolysis, this proceeds via an amino acid active site, as all amino acids do. But what's different, if you remember mechanisms, we talked about mechanisms of different enzymes and the active site residues. The active site residue for phosphoglycerate mutase is actually a phosphorylated histidine. Okay, it's not just a regular old histidine, it's a phosphorylated histidine. Okay, so that is kind of anomalous. Or in the sense it's very rare that the active site is not just an amino acid, but a phosphorylated amino acid. So, okay. So, that mechanism really is a phosphate exchange. That is how phosphorylate mutase functions. You can actually get a 2,3-bis phosphoglycerate intermediate here. Okay, sometimes through this intermediate, some of this leaks off and can go and regulate hemoglobin binding. But basically, this is a mutase. You shift the phosphate, the phosphorylated histidine catalyzes a phosphate interchange, or the inorganic phosphate sort of interchanges and substitutes from the 3 carbon to the 2 carbon. And this histidine, with its phosphorylated group, mediates that exchange. So that's reaction 8 of glycolysis. Let's go to reaction 10 of glycolysis. We take 2-phosphoglycerate, and in reaction 9, we make the high-energy molecule phosphoenol pyruvate. So reaction 9 of glycolysis takes that phosphoglycerate, 2-phosphoglycerate here. All right, so let's draw that. Okay, so here's 2-phosphoglycerate from the previous reaction, and 2-phosphoglycerate is going to get converted to the high-energy molecule phosphoenol pyruvate. So what really essentially happens is that this hydroxyl group is going to leave as a water molecule. Okay, when that hydroxyl group leaves as a water molecule, we have a double bond that forms. Okay. And that double bond forms alongside this phosphate that's attached to this oxygen is kind of unstable. Okay, it's kind of unstable and makes this new bond on the enol pyruvate very labile. So let's begin by drawing the structure of phosphoenol pyruvate. The structure of phosphoenol pyruvate is this. So this comes off as water, as catalyzed by an enolase. And now we have a double bond. This carbon now is attached to this phosphate, but it is now a labile bond, or a hostile, unstable bond, unsteady bond. And this carboxylate stays the same. And this molecule here is called phosphoenol pyruvate. All right. This is reaction 9 of glycolysis. And then finally, we take this high-energy phosphoenol pyruvate. As we stated before, this bond is ready to leave. This phosphate is a good leaving group. And because it's a good leaving group, it is a high-energy molecule. Sometimes you may hear the term high group transfer potential. So I'm just throwing that out there. So this phosphoenol pyruvate has the capability of transferring that phosphate or high energy molecule. Okay, so that's reaction 9 of glycolysis. The final reaction of glycolysis is to take that final phosphoenol pyruvate and give off that phosphate that is itching to get off, and it will give it off and donate it to ADP to give you ATP. And so that is our second ATP molecule that's formed, but you will also get the you'll also get the alpha keto acid pyruvate. So phosphoenol pyruvate from reaction 9 is going to go on to our final reaction. So this reaction actually proceeds in one direction. Okay, it's going to take ADP. This phosphate is going to donate or get donated to ADP. Don't forget magnesium is all around here. Okay, to chelate those negatively charged phosphate groups. And you're going to get ATP. That's a good thing. But you'll also get the final reaction in glycolysis, and that is pyruvate. Okay, the enzyme that catalyzes reaction 10 is called pyruvate kinase. So this carboxylate went here. This carbonyl group went here, and this CH2 group went here. Okay, it proceeds via an enol intermediate. That is the mechanism for pyruvate kinase, and henceforth that CH2 becomes CH3. Okay. So pyruvate kinase goes in one direction. It is exergonic because this is a high energy molecule. That phosphate's primed and ready to go, and it can donate it to a happy recipient in the form of ADP to give you ATP, within itself a high energy molecule, but this is really a high energy molecule because it donates its phosphate to make ATP. This is called substrate level phosphorylation. So the back reaction, because this enzyme goes in one direction, the back reaction is actually catalyzed by a separate enzyme, and that separate enzyme actually does not take pyruvate and convert it to phosphoenol pyruvate. It actually takes oxaloacetate from the TCA cycle and then gets CO2 and uses energy in the form of GTP, and then that generates phosphoenol pyruvate. The enzyme that does this is called PepsiK, or phosphoenol pyruvate carboxykinase. When we study gluconeogenesis, we'll go more into detail of how this TCA cycle intermediate, the citric acid cycle intermediate, starts and kicks off or initiates the gluconeogenic process. Okay, what we talked about basically is the glycolytic process, breaking down glucose. Well, we can just, as well as we go in this direction, we can go in the opposite direction and biosynthesize glucose. That's gluconeogenesis. A lot of these reactions are actually back and forth reactions. Notice there's a back and forth reaction. Okay, back and forth reaction. So other than a couple of enzymes, gluconeogenesis, the building of glucose, is pretty much similar to glycolysis, the breakdown of glucose. They actually proceed twice because you make one molecule of glyceraldehyde 3-phosphate that goes through 6, 7, 8, 9, 10. But reaction 5 actually gives you 2 molecules of glyceraldehyde 3-phosphate dehydrogenase. So you got to go through 7, 8, 9, 10. You got to go through it twice because the Triose phosphate isomerase enzyme here gives you 2 molecules of glyceraldehyde 3-phosphate. So instead of making here 1 molecule of ATP, you actually made 2 molecules of ATP. And instead of making 1 molecule of pyruvate, you made 2 molecules of pyruvate. All right. So when you do the ledger, when you count the number of ATPs that are actually generated, you'll get a net of glycolysis. How many ATPs will you get? Okay, you should get 2 ATPs. Okay, remember we took 2 ATPs away from the hexokinase step and also the phosphofructokinase step, but we made 2 ATPs in pyruvate kinase, and we also made 2 ATPs in the reaction that took 1,3-bisphosphoglycerate and converted it to 3-phosphoglycerate. The enzyme is Phosphoglycerate Kinase. So 4 ATPs, okay, one from pyruvate kinase and the other one from phosphoglycerate kinase, reaction 7, and the last reaction produced ATP. They produce 4 ATPs, but we have to subtract 2 ATPs. Okay, we subtract 1 ATP from hexokinase, and then we subtract the other ATP from where? All right, we subtract the other ATP from phosphofructokinase, the rate limiting step or the rate determining step in glycolysis. So we make 2 ATPs, we also make 2 NADHs. Okay. So another important point before we conclude this, now that we went over glycolysis step by step, molecule by molecule, so it's extremely powerful what we went through together. The next thing is, what does the cell do with this pyruvate? Okay, that is a very important question to ask. Very briefly, basically, it can convert that pyruvate to lactate, and you have generated that NAD+, which can go into and recycled back into glycolysis. So that NADH can go into NAD+. You can take that pyruvate and convert it to ethanol. So here that NADH goes to NAD+, and this can go and get recycled back into glycolysis. Or that pyruvate can go into the TCA cycle and the electron transport chain. And this is what a lot of aerobic organisms employ to maximize their ATP output. But for these guys, mostly bacteria, that generate lactate or ethanol, okay, it's important to realize that this NAD+ that's generated, okay, gets recycled back into glycolysis. And it really gets recycled back at the GAPDH step, the glyceraldehyde 3-phosphate dehydrogenase. Okay, the enzyme that converts glyceraldehyde 3-phosphate, converting G3P to 1,3-bisphosphoglycerate through this enzyme. Okay, so video is on the pyruvate dehydrogenase multi-enzyme complex. So that tells you the first step of how we generate pyruvate into the molecule that's going to feed into the TCA cycle. All right, so this is glycolysis, one of the more fundamental metabolic pathways. Lots of themes in glycolysis in terms of shunting, regulation, and flux. And one final thing, glycolysis occurs in the cytoplasm. Okay, so in metabolism, we also want to concern ourselves with localization in the cell, where it's occurring. And in this particular pathway, glycolysis is occurring in the cytoplasm. We're going to contrast that with other metabolic pathways that occur in other parts of the cell. So that's glycolysis, all 10 reactions. We went through structure by structure, and the ATPs generated by substrate level phosphorylation, the high energy intermediates. So I hope you get something from this, and if you follow this pathway through, from beginning to end, I think you'll have a very, very strong hold of what's happening in glycolysis and where the atoms are going and where the phosphates are going to generate ATP. Okay, thank you.